changes at the tissue-specific level. But in the past, we could not because simply we do not have access to these tissues here. And therefore, traditionally, people have used patients' blood or patients' serum as indirect biomarkers of heart disease. And I think, this is actually not, when you think about it, this actually doesn't make that much sense, right? So what's the point of studying patients' blood to understand heart disease, studying patients' blood samples to look at brain disease, or studying patients' heart samples to look at, blood samples to look at liver disease and so forth. And I would say this has not changed with the human iPS cell model. The human iPS cell model, while it's not exactly the same as the human heart, it's probably the best technology that we have that integrates the patient's DNA, the RNA, the transcriptomics, the proteomics, the metabolomics, and it's all encapsulated in this iPS cell technology right here. I'll explain why here. So the iPS cells, as many of you are aware, was first discovered by Shinya Yamanaka back in 2006 in mouse, and in 2007 in human model. He received the Nobel Prize in 2012 for this discovery. These cells are essentially the same as the human iPS cells in terms of pluripotency, in terms of self-renewal properties. So the way we make iPS cells is that we can take skin or blood or urine samples, even hair samples from patients, and then we culture these cells, and we hit them with 4 reprogramming genes using different methods as shown here. And then with these reprogramming genes, we can then transform the human somatic cells into iPS cells, which for all practical purposes are the same as human embryonic stem cells. Now, once you're able to generate these human iPS cells, through better understanding of developmental biology, you should be able to differentiate these human iPS cells into heart cells, brain cells, liver cells, endothelial cells, and so forth. And once you're able to generate patient-specific as well as disease-specific heart cells, endothelial cells, and so forth, you should be able to come up with comprehensive drug testing. In the case of cardiac, test the contractility of the cells, test the morphology, the arrhythmias, the metabolisms, and so forth here. So when you think about this technology, you should be able to recruit patients with specific cardiovascular disease and try to do disease modeling. And once you're able to collect enough of these patients, you should be able to do RNA sequencing on these cardiomyocytes to understand the pathways, the interactomes, and the cellular circuits. So that allows you to do drug discovery. You should be able to recruit large sample size and essentially trying to do a clinical trial in a dish. On a single patient level, you should be able to do personalized, predictive, and preemptive, as well as quote-unquote precision medicine, which I'll talk about. And lastly, you should be able to take patients who have either brain disease or heart disease and generate their own patient-specific brain cells and heart cells and do them for regenerative medicine applications. Although this is going to take significantly much longer period of time because of all the complexities that are involved in cell therapy, as well as the market models for doing personalized cell therapy. And because of time, I won't get into this aspect of cell transplantation with today's talk. Now, I want to talk to you about the production of cardiomyocytes from ES cells, from iPS cells. So we've been working on this for a long period of time. And for this audience here, I just want to tell you that the technology has improved dramatically over the past 10 to 12 years. We started out with the embryo body differentiation. We now move to chemically defined differentiation and chemically defined feeder-free differentiation as shown by panel C here. A lot of the work in our lab is trying to figure out what are the different factors that you can add to push them into better cardiomyocytes, more mature cardiomyocytes, and also lineage-specific cardiomyocytes. And here is a protocol showing that you can add, for example, activin A, BMP4, and you can add cardiac specification factors as shown here, and then you can push them into cardiomyocytes here. Now, more recently, we've also developed what we call chemically defined generation of human cardiomyocytes. And for this one right here, we use a protocol, a media that has basal media, L-ascorbic acid, and human recombinant albumin. We call this a CDM3 media. The advantage of this media is that we only use 3 compounds. We avoid the use of FBS, a fetal bovine serum. FBS is a xenogenic product, and this could affect bat-to-bat variability of your cardiomyocyte differentiation. In addition, if you're thinking about clinical trials, so you try to avoid as much as xenogenic product as possible. Finally, with this CDM3 media, it allows us to each bottle of this CDM3 media typically costs us around $50 to $60 to make in our lab, which is a dramatic cost saving compared to if you were to buy, for example, E8 or NT media, which would cost you anywhere from $200 to $300 per bottle. So this is a significant cost saving for us because we're making a lot of these cardiomyocytes both for our lab and also for distribution to outside collaborators. So back in 2004, when I started my lab, we were working mainly with human EA cells, and for these human EA cells, our differentiation efficiency was about 5%. So you can see on this video here, there are some cells that twitch on the periphery, but mostly these are cells, a majority of these cells are not twitching, and so these are non-cardiomyocytes. And fast forward about 12 years later, instead of using human EA cells, we routinely collect blood samples from patients. From these blood, we collect the PBMC. From the PBMC, we reprogram them into iPS cells. From the iPS cells, we differentiate into beating cardiomyocytes. We're getting consistently 90% plus beating cardiomyocytes as shown on this video here. Now, working with our collaborators, for example, Beth Pruitt at Stanford, we've been doing 2D micropatterning, in which we can, this is a 7-to-1 ratio. We can see the nucleus of the cell as well as the nice sarcomere structure of a single cardiomyocyte right here. Working with Wolfram Zimmermann in Germany, we've been doing 3D tissue engineering of these human iPS cardiomyocytes. Essentially, here is a collection of these cardiomyocytes. They're attached to 2 posts, and these cardiomyocytes, when they contract, you can see the movement of the post. This allows us to measure the systolic force, the diastolic relaxation, as well as the arrhythmia propensity of these iPS cardiomyocytes before and after different kinds of drug treatment. Now, keep in mind that these human iPS cardiomyocytes are not exactly the same as adult cardiomyocytes. So shown here on this slide, you can see that in terms of morphology, these cells are round compared to rod shape. These cells are these sarcomere structure, unless they're aligned using the 2D micropatterning. By themselves, they're disorganized compared to polarized right here. The action potential is also different. The resting membrane potential is -60 compared to -90. This is due to the presence of the funny current right here. The sodium channel is also less compared to the adult cardiomyocytes. The conduction velocity is slower compared to adult cardiomyocytes. Calcium handling is it's inefficient compared to the adult cardiomyocytes. Mitochondrial bioenergetics is low compared to the adult cardiomyocytes and so forth. So at the end of the day, I would say that, again, these cells are not the same as adult cardiomyocytes. The question is which camp are you on? If you're on the glass half full camp, you say that this is good enough for us to study, and there's always room for improvement, as shown by the 12 years of work that we've been working on this, in which we can significantly improve the efficiency of the cardiomyocyte differentiation. If you're on the more pessimistic side, you say that the glass is half empty. They're not exactly the same, and therefore it's not worthwhile pursuing. Now, obviously, we're on this camp, and we're on the half full camp, and so our entire lab is dedicated toward how do you improve these cardiomyocytes, how do you mature them, how do you make lineage-specific cardiomyocytes, and how do you use these cells for disease modeling, for drug discovery, and eventually for potential cell therapy in the future. So I want to cover some of the aspects of the disease modeling next. So shown here is a cardiomyocyte, and within the cardiomyocyte, there could be various mutations that could happen to our patients. For example, lung QT diseases here. CPVT, ARVD, hypertrophic cardiomyopathy, and dilated cardiomyopathy as shown here. Now, I want to give you a clinical context here. So the clinical context is trying to use this integration of a genotype-phenotype iPSL platform to solve a clinical conundrum. So this is a patient family that was referred to us, in which the mother, as shown by the middle panel here, presented with arrhythmia, and on the MRI had sagittal. On the MRI had hypertrophy of the heart, shown on the left-hand panel. And it turns out that two of her kids already have the phenotype, and the phenotype again is shown by shown by cardiac hypertrophy as well as cardiac arrhythmia. And because it runs in this family, we decided to do DNA sequencing. And the DNA sequencing shows the myosin heavy chain 7 with alternating histidine switch as shown here. Now, kid number 3 and kid number 8 are also genotype positive but phenotype negative. So we were asked by our referring physician and the patients, how does having this myosin heavy chain 7 mutation cause cardiac arrhythmia and cause hypertrophic heart in these patients? And how do we know for sure that this is the actual mutation that caused the phenotype? Because as you know, when you do DNA sequencing, you may come up with several hits, several potential variants of mutation that could explain the phenotype. And so just a little bit background about cardiac hypertrophy. This is the most inherited cardiovascular disease. Affects 1 in 500 people. This is one of the most common causes of sudden cardiac death in young adults. This is a diagnosis of exclusion for cardiologists when we see or hear a young kid who passed out in the football field, basketball court. Back in 1989, first gene identified mutation in myosin heavy chain 7. Since then, more than 1,000 mutations identified. This is the echocardiogram. You can see how thick the muscles are right here on various cuts, sagittal cuts, and also apical cuts right here. And so we generated the iPSL cardiomyocytes for the entire family, and this is a control siblings compared to the hypertrophic siblings. And you can see on a single cell level, the hypertrophic sibling already have much bigger cell size compared to the control siblings. On a single, when we do the patch clamp as well as multi-electrode array analysis, we can see the hypertrophic siblings also have much more delayed after depolarization as shown here compared to the control. The DADs are essentially a phenotype in which if you have extra beats, for example, on the QT interval, and then the extra beat can actually cause VT, VFib, and even torsade. So this is not a good sign for these hypertrophic siblings. And so you can aggravate the hypertrophy as well as the arrhythmia by giving these cells isoproterenol. You can block it by giving these cells verapamil and propranolol. This is a calcium channel blocker, and propranolol is a beta blocker to reverse hypertrophic and arrhythmia phenotype. So we spent about 2 years studying this family, and to make a very long story short, what we think is happening is that the mutation, myosin heavy chain 7 mutation, causes increase in cellular calcium, leads to activation of the NFAT signaling pathway, leads to hypertrophic gene response. Calcium handling defects lead to arrhythmia, and you can exacerbate the arrhythmia and hypertrophy by giving them isoproterenol. You can block it by giving them a beta blocker, for example, propranolol. You can also block the arrhythmia by giving these cells lidocaine, mesilazine, ranolazine. And you can block the initial step by giving them verapamil. So this is one example in which you have a patient family that comes in with a particular disease. The referring doctors are asking you, what is the exact mechanism? It's not possible for us to biopsy the patient's heart, but it's okay for us to biopsy the patient's skin or take the blood, make iPSL, differentiate the cardiomyocytes, study them, and understand what exactly is the pathway that causes this particular disease phenotype. Now, the other patient family that I want to just show you or highlight is this particular family, which we also study. It's a familial dilated cardiomyopathy in which this kid here already had a heart transplant at age 15. when you go back to the family history, you'll realize that the father, as well as the uncle, as well as the grandmother, all of them had dilated cardiomyopathy. So this runs in the family, so it's familial. We decided to do DNA sequencing. It's a troponin T mutation with the arginine to tryptophan switch right here. And again, just like the previous family, we're trying to figure out how does this particular troponin T mutation cause a dilated cardiomyopathy phenotype. And so we're able to generate iPSL cardiomyocytes from an entire family cohort, able to show that the diseased patients with the DCM phenotype had altered regulation of the calcium ion, decreased contractility, abnormal distribution of the sarcomere alpha-actinin. And then you can treat it with the beta block, with the beta adrenergic agonist, isopaterol, to worsen the phenotype, and you can treat it with the beta blocker, metoprolol, to improve the phenotype. Recapitulating results from multiple large beta blocker clinical trials. Now, keep in mind that once we make these iPSL, they are essentially with us, for the next 5, 10, 20 years. Essentially, these cells, as I mentioned before, they're pluripotent, they can self-renew. We can pull out these cells and study them a couple of years later if we want to have a deeper analysis of these cells. This is very different from clinical trial in which patients could come in and then afterwards they could move out of the particular location, in this case Bay Area. So it's not possible, no longer possible for us to study these patients. These cells, once we bake them, we can pull them out, we can study them again. So this is another study on the same dilated cardiomyopathy patient cohort in which one of our postdocs was able to show that the mutant troponin T actually translocates into the nucleus, binds to histone demethylase enzymes KDM1A and KDM5A, causes epigenetic changes so that there's increased phosphodiesterase transcription, which increases phosphodiesterase 3A as well as phosphodiesterase 2A. Both of them causes increased hydrolysis of the cyclic AMP, which decreases protein kinase A, which causes decreased calcium transient and decreased contractility. So in the end, we're able to come up with a additional mechanistic explanation for the family that is epigenetic regulation of the phosphodiesterase 2A and 3A causes compromised beta adrenergic signaling in these iPSL models of dilated cardiomyopathy. Now, I've mentioned both the hypertroph and the dilated cardiomyopathy. The dilated cardiomyopathy is perhaps the most common cause of heart transplant in young adults. The third most common cause of cardiomyopathy is left ventricular non-compaction. This occurs quite often in pediatric heart disease here. And so a couple of years ago, we were fortunate enough to recruit a large family at Stanford, another smaller family in Japan. And this large family at Stanford, this kid here already had a heart transplant, as shown by the black box right here. Two of his siblings also has LV non-compaction, but they were asymptomatic. The father had the mild dilated cardiomyopathy. This kid who had heart transplant, you can see the explanted heart has this spongiform formation. Basically, this hypertrophic in the heart. This is bad because it could impair the cardiac contractility. It could also cause blood clots to form in these crevices and cause a stroke to the patient. When we did the DNA sequencing, we found that it was TBX20. One of them had mutation at the T-box domain. The other one had mutation at the transactivating domain. When we make the iPSL, we can show that at the early stage, the iPSL cardiomyocytes, when the LV non-compaction patients are hypoproliferative compared to the control patients. When we genome edit out the mutation, or correct the mutation, we can see we can rescue the proliferative capacity of these early cardiomyocytes. Again, the early cardiomyocytes can proliferate. The later cardiomyocytes will not be able to proliferate. When we did the RNA sequencing, we found that the TGF-beta is overly expressed in these LV non-compaction cardiomyocytes. So for this particular family, what we think is happening is that the TBX20 mutation causes increased TGF-beta. Increased TGF-beta causes increased cell cycle regulators. increased alpha cycle regulators impaired ventricular myocardial specification and growth, and in the end causing the hyperregulation and causing the alveolar compaction phenotype. So this is also, in our opinion, very satisfying for the family, for them to understand how this particular mutation causes the phenotype there. And so more recently, in collaboration with Jeff Mokenton's group, he was able to actually generate a series of cardiac-specific mouse models that allows him to do systemic fine-tuning of the sarcomere tension, as shown here, as well as the sarcomeric reticulum release of the calcium, and then this causes either hypertrophic heart or dilated heart. And these data were also confirmed using human iPSL cardiomyocytes that were derived from the hypertrophic patient that I showed you earlier with the myosin heavy chain 7 mutation, as well as the dilated cardiomyopathy patients that I showed you earlier with the troponin T mutations. And so far, what I've shown you is mostly using this particular model to do disease modeling in hypertroph, in dilated, in alveolar compaction. You can also do this in patients with long QT disease, and also using genome editing. So in this particular case here, we have a healthy patient. We have a long QT1 patient that has prolonged action potential. And we took a healthy patient and genome edited it with the long QT1 mutation. We can see prolonged action potential as shown here. So this is important because this allows us to bypass patient recruitment, because we are limited by the number of patients that are referred to us by our physicians. And if the patients do not have that particular family, if patients do not have that particular mutation, we cannot study them. So here with the genome editing, CRISPR or TALEN editing, we can then create these custom designed IPS lines with their various mutations. And then this is important because from a clinical perspective, we should be able to combine genome editing iPS cells to assess patients with particular variants and certain significance as shown here. So you may run into examples in which a family member is referred to your clinic, and the patient, for example, had a history of syncope, and you do an EKG, and the EKG shows a prolonged action potential, a prolonged QT interval, maybe about 480. It's prolonged, but it's not, overly prolonged. And so you send it out for GeneDx testing. It came back as a VUS, a variant of uncertain significance. So in this case, you're kind of stuck. You're stuck in the sense that you don't know exactly what to do with the patient. You'll probably ask the patient to come back next year, the year after that, and do EKG again, ask the patient if he or she has passed out in the interim. This is very difficult for us as a clinician because it's not satisfying. It's not very satisfying for our patients to kind of follow them all the time without giving a definitive diagnosis. So what I'm trying to say here in this slide is it possible to make iPS cell cardiomyocytes and show that there's arrhythmia in these cardiomyocytes? Is it possible to genome edit out the VUS to show the arrhythmia goes away? Is it possible to genome edit in the VUS into a healthy, normal iPS line to show that the arrhythmia pop up? And once you have these additional confirmation, is it possible to tell the patient that, yes, this is actually a pathogenic VUS, and you should be much more vigilant about this, and we should be much more aggressive in terms of medical treatment? So we don't have this data to back this up. I mean, this is something that we're interested in doing the next couple of years to further demonstrate whether this is possible or not. And so just to summarize, I think right now, for example, the traditional paradigm is that you have patients that refer to you, and for example, in this case, 2 of the kids may have a disease, another kid is a questionable mark, and right now we just follow this entire family over a long period of time. It's very slow, it's very ineffective. Question is, in the future, are you able to take this kid right here that has a known diagnosis or has not manifested the disease, the phenotype yet, make the iPS cell and generate the cardiomyocytes, and able to predict whether the patient is going to have disease development or progression in the future. So I want to shift gear to the third part of my talk, which is how we use the genotype-phenotype iPSL platform for precision cardiovascular medicine. So this is an interesting slide because it shows the top 10 medications that are being used in the U.S. And for each medication in which it helps, as shown by blue, there are anywhere from 3 to 24 patients in which they take the same medication and has no effect. For example, if you take Humira, 1 out of 4 patients would have a positive effect. And if you take Nexium, 1 out of every 24 patients would have no effect, would have a positive effect. So this is actually not very good, and that's why the article, the title is "It's a Time for a One-Person Clinical Trial." So I want to give you the example of how we do this in terms of iPSL cardiomyocytes and cardiac oncology. So the example is breast cancer. This is the second leading cancer death in women with 12 million cancer survivors in the U.S. 8 to 10% of the women will experience doxorubicin-induced cardiac toxicity acutely. Among the survivors, 20% would develop heart failure over 5 years, and 40% would develop heart failure if there's a combination of doxorubicin, Herceptin, and more than 75-year-old. So the current treatment guideline for cardiologists is to give beta blocker plus or minus ACE inhibitor once the echocardiogram shows reduced LVEF. So this is actually not very satisfying as well, right? So the patient presented with the manifestation of reduced LVEF. You then start giving the medication. So the question here is this considered cardiovascular precision medicine? And can we do better than this? And so more recently, we did a study in which we recruited breast cancer patients that had treated with doxorubicin. A year later, some of them had normal cardiac function. Some of them had doxorubicin-induced cardiomyopathy. We make iPSL differentiate into cardiomyocyte, subject them to battery assays, for example, contractility assay, calcium cycling metabolism, gene expression, apoptosis, and chemoprotection assays. And then we're trying to predict which one of these patients will develop cardiac disease and which one will not. And so in this particular study right here, 4 healthy patients, 4 breast cancer patients who had doxorubicin but did not develop cardiac toxicity, we call them docs. 4 of them did develop cardiac toxicity, we call them dox-tox. For example, here you have a 31-year-old woman, her ejection fraction went from 70 down to 45. And likewise, you have a 40-year-old woman, her ejection fraction went from 77 down to 36. Now, when we make the iPSL cardiomyocytes and expose them to doxorubicin, for example, at 1 micromolar, the dox-tox group have much more sarcomeric disarray compared to the dox. At 1 micromolar, as shown here, the dox-tox group have much more apoptosis compared to the dox group, as well as the healthy patients. At 1 micromolar, the dox-tox group also had much more irregularity of the contraction. Essentially, the cells have stopped contracting compared to the dox group. And then at 1 micromolar, the dox-tox groups also have much more impairment of the viability assay as shown here as well. So here, again, even though we do not know the genetics of what's causing the doxorubicin-induced cardiac toxicity, the iPSL cardiomyocytes encapsulates the DNA, the RNA, the protein, and it's giving a readout. And the readout is that upon exposure to the same level of the doxorubicin, the iPSL cardiomyocyte that came from the dox-tox group, these are the women who took the doxorubicin and then later on developed cardiac toxicity, have much more impaired viability, cellular viability, contraction, and so forth, as shown here. Now, the other thing is once we're able to make iPSL cardiomyocytes from these patients, we should be able to screen for medications that can improve the cardiac contractility and improve the viability of these cells. So this is in collaboration with the local startup company called Stem Cell Diagnostics, in which we're able to show out of screening more than 20 compounds, one of the compound in particular, SC018, when compared to FDA-approved drug, dexazosane, it prevents cell death in response to doxorubicin. It protects mitochondria against doxorubicin, protects electrical and contraction properties, and also reduces the doxorubicin-induced double-stranded DNA breaks. And then it has no effect on cancer killing. So this is good because we want to protect the heart, and we want the medication not to protect the cancer cells right here. So we're doing additional validation studies on this particular drug because we think that this type of platform will allow us to accelerate the time that's needed for drug repurposing as well. And so I kind of show you examples of doxorubicin. You could also do this with tyrosine kinase inhibitors. These drugs, as you all know, are probably the most prevalently used cancer medications these days for various types of cancer. For example, breast cancer, non-small cell lung cancer, thyroid cancer, melanoma. Problem with these medications is that they are also associated with cardiac toxicity. They could cause tachycardia, congestive heart failure. With Gleevec, for example, they could cause impaired LVEF. They could cause QT interval prolongation. They could cause torsade issues as well. So just like the previous study, we were able to recruit more than 10 patients, make iPS cell, generate the iPS cell cardiomyocytes, treat them with the various TKIs, do RNA sequencing, and then do cardioprotective drug screening, and also do receptor tyrosine kinase phosphorylation panels, and a whole bunch of assays that I haven't shown here. And then this is a typical panel here in which these are the different drugs that were treated on these patients. And what we're trying to do is essentially develop a cardiac safety index for these tyrosine kinase inhibitors that are being used to treat cancer patients. So again, shown here on the left side is the list of the medications. On the right side is the cardiac safety index based on the iPS cell cardiomyocytes. And the right side here is clinically reported cardiac toxicity. So this work will come out in Science Translational Medicine later on. And so I won't go over all the experimental details here. And so what I just want to summarize is that we think these iPS cells can be used for precision cardiovascular medicine. So right now it's patient history, clinical exam, and we give patient different medications. Essentially the patients are guinea pigs, and we think this is slow and ineffective. And the question is, in the future, is it possible to take various cell types, brain cells, heart cells, liver cells, kidney cells, do drug screening, come up with the best medication that the patient should be taking, and give it to the patient here. Now I want to shift gear to the last part of my talk, which is trying to integrate the genotype-phenotype iPSL platform for drug development. So this slide is very interesting because essentially it's called the Erins Law, and it says that the efficiency of research and drug development in the U.S. Decreased by half every 9 years. And so this is Erins Law. It's a cynical way of the Moore's Law, which is that the amount of microprocessors increase double every 18 to 24 months right here. And what part of the issue is that the authors think drug repurposing could help counter this decline, because drug repurposing typically costs about $300 million and takes about 6 years, whereas a new drug would take about 12 to 16 years and $1 to $2 billion. And a lot of folks are thinking about how do you incorporate the iPSL model for drug discovery and for drug repurposing. So this is a review article by the R&D folks at Pfizer, in which the concept is taking a lot of patients, make iPS cells, and differentiate them to brain cells, heart cells, liver cells, adipocytes, endothelial cells, hematopoietic cells, come up with the assays, and I've kind of shown you assays of cardiac earlier, contractility assay, arrhythmia assay, and single cell assay, and then compare cohorts. Cohorts means the difference between patients who are healthy versus patients who have cardiovascular disease, difference between women and men, difference among Caucasians, Asians, Hispanics, African Americans, and so forth right here. And so I just want to show a couple examples of what we've been doing. This slide shows the drugs that have been withdrawn due to cardiac toxicity, some of them due to poor arrhythmia, some of them due to heart attack, some of them due to QT interval prolongation issues. I want to point out cisapride or propulsant here, because this medication was withdrawn in 2000 right here because of QT interval prolongation issues. Now, we can't give cisapride back to patients because the medications have already been withdrawn from the market, but we can give cisapride to our patients, iPSL cardiomyocytes. We can do a clinical trial in this case right here. So we recruited 12 patients, 3 of them controlled, 3 of them long QT, 3 of them hypertrophic, 3 of them dilated cardiomyopathy. And we did a single-cell patch clamp, and we're able to show that the long QT patient have prolonged action potential, which is not surprising because they have long QT1 to begin with. And when we expose them to low-dose cisapride, all these patients did pretty well. And when we expose them to high dose of cisapride, all of them develop arrhythmia. When we expose them to more physiologic cisapride, it turns out that the long control patients and the DCM patients are fine. The long QT patients and the hypertrophic patients, they develop arrhythmia. So this is very interesting to us because it begs the question, why is it that if you go back to the registry, it turns out the long QT, the torsade, develop in 1 out of every about 30 million patients who took the medication. And why is it that only 1 out of 30 million patients had this type of drug adverse events? And is it really true that healthy, normal patients, which is most of us, if we were to take cisapride, we would develop arrhythmia? I think this data argues that healthy patients should be fine. It's only patients who have genetic susceptibility. For example, the long QT patient, the hypertrophic patients, they have an increased propensity to arrhythmia. And perhaps, in the setting of hypokalemia, in the setting of fever, and the setting of taking the cisapride with genetic predisposition, then they develop this type of trouble. So I think, having this type of information is, in my opinion, would be very helpful for drug companies when they see this type of information during the pre-IND phase. They may decide to just kill the drug completely, or they may decide to reformulate the drug so that this type of arrhythmia disappears in long QT and hypertrophic patients. Or they may decide to push the medication and have some kind of black box warning and say that the patients have an EKG check before they have the medication given to them. So this is an example why we think having platforms, and especially patients with the various diseases, would be helpful. The other thing that we're going after is patients with different ethnicity and phenotype. In this case here, we recruited patients with Asian American patients with aldehyde dehydrogenase 2 genetic polymorphism. This genetic polymorphism explains for why a lot of Asians, when they drink alcohol, they have facial flushing, they have tachycardia. The other problem is that in epidemiology studies, when they get heart attack, they tend to have worse outcome compared to patients who can drink. And this is shown by iPSL cardiomyocyte model, in which this is wild type patients. These are the guys who can drink. These are the ones with aldehyde dehydrogenase genetic polymorphism. And when we expose them to hypoxia, we can see that these cells die much more. And the main reason is that the enzyme is involved in detoxifying alcohol. It's also involved in detoxifying ROS, a reactive oxygen species. Now you can imagine that when you get a heart attack, you're going to have tons of ROS being activated, and lacking this enzyme would put you at a disadvantage compared to the patients who have this particular enzyme. So this is a demonstration using iPSL cardiomyocytes to show major ethnic differences, in this case, East Asians. What about other cohorts? For example, African Americans who have hypertension, Hispanics who have diabetic vasculopathy, South Asians who have metabolic syndrome, and many others. I think you should be able to use these iPSL cardiomyocytes or endothelial cells to also model these particular diseases as well. Lastly, is it possible to do this iPSL-based clinical trial in a dish? So this is an interesting slide put forth by Shinya Yamanaka, in which what he's proposing is that most of the clinical trials are very, very large, and once the result comes back to be negative, the drug company loses interest, the investors lose interest, and essentially the drug is dead. Now, is it possible that this particular drug actually has a positive signal, but the positive signal is being drowned by a negative signal among the non-responders? And it's possible to use the IPA cell model to sort out which one of these patients is going to respond and which one of these patients not going to respond. And once you figure out the responders versus the non-responders using these IPA cells as surrogates, then for your phase 1 or phase 2 clinical trial, you purposely recruit the responders and give them the medication so that you have a higher hit rate, and so that the drug goes to the next phase of clinical testing. The investors do not lose interest. And more importantly, once you understand why patients respond and not respond, you can come up with additional mechanistic understanding and later on gradually expand the indication as you understand the drug better. So a lot of folks are trying to push this concept, including our group. And just to summarize, we recently published a paper in which the title of the paper is Transcriptomic Profiling of Patient-Specific Human iPS cell cardiomyocyte to Predict Individual Drug Safety and Efficacy Responses in Vitro. And here we're able to show that we take patients and we're able to show that reprogramming as well as the cardiac differentiation preserve patient-specific gene responses. Drug-induced cardiac toxicity, in this case here we tested a medication, rosiglitazone, and also another medication, tacrolimus. We were able to show that the drug-induced cardiac toxicity can be functionally evaluated in vitro using patient and disease-specific iPS cell cardiomyocytes. We're able to show that the bioinformatics analysis can be used to help us predict and risk stratify which patient is going to improve and which patient is not going to improve with these medications here. So just to summarize the last part, this is a traditional paradigm in terms of clinical trial model, using transgenic cell lines, using small animals, using large animals, costly, slow, low predictive value. And then in the future, is it possible to add? We're not advocating to replace them. We're advocating to add them so that you take cells from patients of diverse ethnicity, gender, cardiovascular history. You expose these cells to the medications, and then you figure out which medication is going to improve and which one is not going to improve. Gives you increased confidence prior to costly clinical trials right here. And so this is what we're doing at Stanford. We're creating a biobank of about 1,000 patients. So far, we have about 400 patients. And these patients have different cardiovascular history, ethnicity, sex, and also creating isogenic lines using CRISPR genome editing. We're trying to perform DNA sequencing on the iPS cells and RNA sequencing on the iPS cell cardiomyocytes before and after drug response. We're working with Russ Altman to link it to the PharmGK database so that we're trying to understand how human genetic variation impacts the drug response. We link it to the medical information database here at Stanford, and we're working with NHLBI as well as CERM and iPSL biobanking, and with FDA and drug safety testing. And we've established a very robust sharing resource plan with many investigators here. And so essentially, if you're interested, you can email me. We can send you bios of these beating iPSL cardiomyocytes, for example, from normal patients, from DCM, dilated cardiomyopathy patients, hypertrophic patients, long QT patients, free of cost. If you have patients with cardiovascular history with an interesting phenotype or interesting mutation that we do not have, you can send us the blood. We will make the iPS cells. We will bank the cells here. We'll also send the iPS cells back to you. And again, this is free of cost because it's funded by a NHLBI R24 mechanism. So over the past 3 years, we sent out cells to more than 120 groups, investigators in the U.S. as well as across the globe as well. And so I just want to summarize the talk. We think the iPS cells, as shown here, we can use these iPS cells for disease modeling. I've shown examples for hypertrophic, dilated, long QT, albumin compaction. We can use this for personalized medicine at a single patient level. And then on a cohort of patients, we can do this for drug discovery as well. I didn't have time to go into the cell therapy aspect, which we also have is pretty robust activity here at Stanford. And with that, I want to thank the postdoc fellows in my lab, as well as their funding support, the cardiology fellows, the lab instructors, the graduate students, the biobank, the collaborators at Stanford, the collaborators at outside of Stanford, funding support from CERN, from the NIH, from the American Heart Association, and from the Stanford Cardiovascular Institute. And thank you very much for your attention. I'll be happy to take any questions.
So I think Amal is going to read any questions that you may have. Please use the chat box to type in questions for Dr. Wu. Amal, are you on mute?
Hi. Can you hear me?
Yeah, so there's a question by Rosali saying, do you share any of the cell lines that you create? Yes, so we do share the cell lines, so we could send you the IPA cells so that you could do the differentiation yourself. That's not a problem. And it depends on, I guess, let me just clarify this. Let's say, for example, hypothetically, you send us blood samples from patients with some kind of interesting disease that we do not have currently at Stanford. You will send us the blood. We will make the IPA cells. We will bank it at Stanford so that it's kind of owned by Stanford, owned by the NIH. And we will send the cells back to you so that it's owned by you. And the patient will sign both consents as well. Now, we will not send the cells to another investigator because because you're the one who gave us the blood samples in the first place. So we will honor that for about a year, meaning that you can get to work on the IPA cells that we generated for you for about a year. A year later, if somebody asks us for that particular line, then we will send it out to the other investigator. So that's one example. The other example would be, for example, we publish on the long QT, on the DCM, on the HCM, on a whole bunch of control patients, and the aldehyde dehydrogenase. In that case, you could send us an email request and say, you would like to have the undifferentiated IPS lines, and we will be happy to send you the lines. So there's a question by Dan. The question is there heightened concerns for patient privacy when conducting this work? So that's a very important question because, when we first got started with this biobanking, and also with the IPA cells, because it's so similar to EA cells, there's a patient concern about privacy and so forth. So it turns out that we have this all vetted by the NIH and also vetted by the Stanford IRB, by the Stanford School. And the patient signed consent is anonymous consent. And so it's all de-identified, and the patient has already given the consent that we can use this IPA cells, and we can differentiate these IPA cells. We can also distribute these IPA cells to our collaborators. And so this is fine with NIH. This is fine with the Stanford IRB.
Are there any other questions for Dr. Wu? So type it into the chat box.
Yeah, so the question by Amal is that the timing of obtaining patient blood, generating the iPS cells, and getting results in a timely fashion for treatment of patients with fast-growing malignancies or other conditions, is that going to be an issue? And I think Amal is spot on. So for some of the diseases, for example, glioblastoma or pancreatic cancer, it's going to be an issue because it's so aggressive, and the first priority is try to treat the patients as soon as possible. And then, so in that case, I think the current model of making iPS cells, generating cardiomyocytes, making the iPS cells probably about 4 to 6 weeks. Generating the cardiomyocytes is another 4 to 6 weeks. That's not going to help our patients. So the current model is used for diseases that are much more chronic. For example, we picked the example of a breast cancer disease. I think it's possible that in the future, the timeline for making the iPS cells and cardiomyocytes will shrink as people come up with better protocols or additional genes that can shrink the amount of time that's needed. It's also possible to think of this as another paradigm that, if you fast forward 20 years into the future or 30 years into the future, is it possible that kids who are born would have their DNA sequenced? But as I mentioned, if you just look at the DNA sequencing information by itself, it's very junky. It's very hard to figure out what's going on. And is it possible that kids in the future we would just take the umbilical, we take the kids' blood, make the iPS cells, and expose the iPS cells to maybe 20 or 30 common medications that the patient's going to take, and tell them how they're going to respond. And in addition, since we already have the cells, if the patient do develop some type of cancer in the future, to do this type of platform to predict which chemotherapy is going to help or not. So I'm just giving you additional possibilities in the future that this may work. But as of 2016, if you have aggressive cancer like pancreatic cancer, GBM, yeah, it's not going to help.
Okay. If there are no further questions, we're currently at the top of the hour, so I'd like to thank all participants for joining this web conference, and also Dr. Wu for giving a fantastic presentation. Thanks, everyone.
Thank you very much. Bye-bye.